With regard to this yesterday problem, message yesterday problem, you message yesterday problem, that is 399 amino acids, how many are? Okay. How many? Yesterday problem, how many amino acids? The problem. Are you not getting it? Yesterday, how many amino acids you said? 399 or 400? Usman, Sheikh Sultan, 199 amino acids. Okay. So, with regard to the problem, number of amino acids in polypeptide, 199 amino acids. Okay, it is 199 amino acids. Okay. In 199 amino acids, suppose amino acid 1, amino acid 2, like this it is 199. So it is polypeptide. It is polypeptide. On mRNA, that is 5, 3 mRNA. How many codes? 199 codons plus nonsense codon. 199 amino acids, 199 codon plus 1 nonsense codon. 199 plus 1, 200 codons. Number of codons, 200. 1 codon, 3 nucleotides. 200 codons, 600 nitrogen bases. 600 nitrogen bases. So, in this DNA, okay, so on this mRNA it is having 600 nitrogen bases. This is 3, 5, 5, 3, it is DNA. It is having 600 nitrogen bases, 600 nitrogen bases. Okay. And This 3, 5, it is transferred. That means information to this, what it is, mRNA. So it is having what it is 600 nitrogen bases, 200 what it is, the codons. So this DNA, it is having total DNA 1200 nitrogen bases. Length of the DNA, how much? For 20 nitrogen bases, 34 angstroms. 600 plus what it is, 600. 1200. 1200 into 34 by 20. Two not 4 angstrom. So here length of DNA 204 angstroms. Okay. And length of mRNA, length of mRNA. So up to this it is, suppose 34 angstroms transfers how many bases? Only, suppose it is one spiral, 34 angstrom, 10 nitrogen base, 10 nitrogen base. How many are transferred to the DNA? 10. For 34 angstroms, 10 nitrogen bases. Okay. So 10 nitrogen bases, the length it is 34 angstroms, mRNA. 600 nitrogen bases, 600 into 34 by 10. 2040. For 20 nitrogen bases, 34 angstrom. 1200 like this. So 10 nitrogen bases, 34 angstroms transfers for the 10 nitrogen bases to mRNA. 10 nitrogen base, 34 angstroms, 600 nitrogen bases transferred. So 600 into 34 by 10. So 2040 angstrom length of what it is, mRNA. 2040 angstrom length of what it is, mRNA. Okay, the second. How many carbon nitrogen rings? 600 purines, 600 pyrimidines. So next question it is, number of CN rings. 600 into 2, 1200. 600 into 1, 600. 1800. 1800 carbon nitrogen rings. Is it okay? All message it is okay. all Navya once length of the DNA and RNA. Okay, Navya here once DNA for one spiral it is having 34 angstrom. The 34 angstroms having what it is 10 and 10, 20 nitrogen bases. Length of the DNA for 20 nitrogen bases 34 angstroms. For 1200 nitrogen bases, how many angstrom? Okay. In DNA, in total 34 angstroms, how many nitrogen bases transferred to the mRNA? Only 10. This strand only transferred. So, for 10 nitrogen base, the length it is 34 angstrom. For 600 nitrogen bases, how many angstrom? That comes under what it is 2040. Okay. In DNA, two strands 600, 600. Total it is codons 200. One codon, 3 nitrogen bases. 200 into 3, 600. 600 nitrogen bases. So here it is 600. Another 600 in DNA. Total it is 1200. So 600 into 2 plus 600 total comes under what it is 1800 carbon nitrogen rings. Himalakshmi, okay. Venkataswamy, okay. Navya, now okay. Message. Shaikh, okay. Okay. Padma Priya, okay. Vishnu, okay. Anushka, okay. Navya, okay, it is. Okay. So we have to consider what it is nonsense codon, terminating codon, because polypeptide chain is going to set free only after completion of what it translation. Translation is terminated by means of what it is nonsense codon UAA, UAG or UGA. Navya, okay. Now gene regulation important. Now it is gene regulation. Gene regulation. So the DNA having information for what it is particular protein. DNA having information for particular protein. So this transfer to what it is mRNA. So what it is mRNA. mRNA make what it is the protein. mRNA is going to make what it is the protein. So metabolic activities which are controlled by it is protein. So exhibition of what it is trait or phenotype. Certain metabolical activities has to be exhibited, that is by means of what it is enzymes. There is no improper metabolism, improperly it is expressed. Proper metabolism, properly expressed. So enzyme has to be synthesized. So biological information present in nitrogen bases made available in the cell by synthesizing the protein, the phenotype is going to be expressed. Okay. To understand this, genes provide potentiality. It is having the potential. Genes provide potentiality. Environment provide opportunity. Environment provide opportunity. So genes plus environment, exhibition of phenotype. So genes control the exhibition of what it is character by synthesizing what it is proper protein. Okay. So imagine yourself, you are having so anxious to learn many things. You are having such a potentiality, but you are not having such an environment to study. Even though we are having potentiality, we are not supposed to exhibit the full potentiality. Suppose if you have an environment, the full congealed environment, but you are not having that much of the potential, we cannot express. Not only the genes, environment also should be congenial for expression of the character. So genes provide potentiality, environment provide opportunity, ultimately responsible for exhibition of character, that is phenotype. To understand with more clarity, if you take a tall plant, capital T, certainly it is going to be tall. But made to grow in an environment which is not suitable, nutrients are deficient, and temperature is not sufficient, in that it is not going to exhibit the character at all, even though it is having the potential. But environment is not providing such an opportunity. So, genes which are on and off, whenever it is required, it is exhibited. Whenever it is not required, it is not going to be exhibited. What do you call it as gene regulation? Genes which are regulated depends upon what it is requirement. Okay, that's what do you call it is gene regulation. So, biological information present in the nitrogen bases made available in the cell by synthesizing a proper protein. What do you call it? Gene regulation. Genes provide potentiality. Environment provide opportunity. Ultimately responsible for exhibition of character. Okay. Up to this it is okay. Introductory part. All introductory part it is okay. Introductory part it is okay. Okay, Lucy, okay. So here, biological information present in nitrogen bases made available in cell by synthesizing protein called gene regulation. What do you call it? Gene regulation. So genes switched on, switched off. The gene switched on as well as switched off depends upon the requirement. And these genes meant for function or set of functions which may be switched on or it is switched off, depends upon what it is requirement. Okay, what do you call it? A gene regulation. So metabolism activity by means of the enzyme. If proper enzyme, proper metabolism. There is no enzyme, improper metabolism. That character is not going to be exhibited. So for that particular protein, the information present in DNA transferred to mRNA. It makes the protein. Earlier we said, why not DNA not directly involved in transferring this? Why not involved in expression of the character? Because DNA is a long molecule present in nucleus. Amino acids are present in cytoplasm. Ribosomes are available in case of cytoplasm. So every time it cannot move from the nucleus to what it is cytoplasm. Further it is a long molecule held with what it is the protein. So it is making the process easier by transferring it to what it is mRNA. It is the director main and transferring to what it is mRNA, assigning mRNA to proper function by synthesizing what is the proper protein. So mRNA leaves the nucleus, enter into cytoplasm and associate with the ribosomes to make what it is the proteins. In earlier classes we completed the translation, splicing, what it is. Okay. So gene switched on and off. And gene regulation in case of prokaryotes. Regulation means the protein has to be formed. Okay. In prokaryotes, gene regulation occurs at transcriptional level. What it mean? A transcription level, in transcription only what is formed? mRNA is formed. So translation and transcription which are coupled in case of the bacteria. Why? It occurs in one compartment. And translation began even before mRNA is fully transcribed. So, in prokaryote, gene regulation occurs at variety transcription level because transcriptional initiation itself a predominant target site for transcription initiation itself a predominant target site for gene regulation. Transcription initiation itself a target site for gene regulation because transcription and translation are coupled occurs in one compartment. There is no demarcation of nucleoplasm and cytoplasm because in one compartment, no demarcation. Further, mRNA does not need any processing in case of prokaryote. That's why transcription initiation itself a predominant target site for control of what it is the gene. It is regulation, it is expression. For control of what it is gene expression. Because in this mRNA, no need of processing. Earlier we said, why no need of processing? Genes are non-split. Why? Genes are non-split. So it is not visible. mRNA, no need of processing. For this point, genes non-split. This is the point. So in prokaryote, transcription initiation itself a predominant target site for control of RT gene expression. Because it occurs, both are coupled in one compartment, mRNA do not need any processing. Okay. Gene regulation in eukaryotes, it exerted at various level. So gene regulation in eukaryotes is a broad term because it exerts at various levels. That is, transcriptional level, that is formation of transcript. You observe this. There is only one sentence given in case of the text, transcriptional level. How the gene is regulating at transcriptional level. Suppose it is the DNA. Okay, it is the DNA. it is structural gene. It is RNA polymerase. To form mRNA from the structural gene, RNA polymerase has to bind it where it is promoter. RNA bind it where it is promoter. But some other proteins which interacting with the DNA affect RNA polymerase in detecting where it is promoter. Adjacent there is a site. There is some other protein. It is regulatory protein. It binds at this portion. So RNA polymerase do not bind at promoter. There is no transcription of where it is structural gene. So there is no expression. So transcriptional level means RNA polymerase has to bind at where it is the promoter. So regulatory protein affect RNA polymerase in detecting where it is stored sites. It is affecting at transcription level. For this point, do write down, it is regulatory proteins affect RNA polymerase in detecting where it is stored sites. Regulatory protein affect RNA polymerase in detecting stored site. Because some other molecule bind at this DNA, RNA polymerase finds no way to find where it is the promoter to transcribe where it is structural gene. So it affects at a transcription level. Okay. In order to express the gene, it has to cross so many borders. It is the first border, that is transcriptional level. Formation of primary transcript. The primary transcript is formed. The second, it is process level. That is hnRNA to mRNA. hnRNA to mRNA. So, processed RNA having different exonic sequences. In text only one sentence given, processing level. We know processing hnRNA converted into mRNA. So, at exons, processed RNA having different exon sequences. Suppose exon 1 intron, exon 2 intron, exon 3 intron, exon 4 intron, exon 5 intron, exon 6 intron, exon 7. Suppose a protein requires only E1, E4, E7. The other protein, it is having E3, E5, E6, like this. So mRNA with the different exonic sequences are possible because hnRNA with multiple introns are spliced in different ways. In one way, it is going to form this polypeptide. Polypeptide 1, it is polypeptide, another it is polypeptide 3. So try to get the point. In prokaryote, one mRNA, many polypeptides, polycystonic. Here, one mRNA, many polypeptides possible by alternate splicing. That is, mRNA with different exonic sequences are possible because the hnRNA with multiple introns are spliced in different ways. In one way it is found polypeptide, another way it is polypeptide 2. This having E1, E4, E7, this having E3, E5, E6. It is one polypeptide, it is other polypeptide. So, RNA with different exonic sequences. Suppose it is assertion question. RNA with different exonic sequences. Which RNA it is? Mature RNA with different exonic sequences. Can you able to differentiate the primary transcript and mature RNA? It is primary transcript and mature RNA. The primary transcript is going to have what it is exons and intron. And processed RNA is going to have only exons without what it is introns. Have you got the point? Okay. Okay, are you getting the point? Reddy. Okay. It is very, very important. Because text only sentence given, we have to elaborate this, what it is the matter. Likitha, okay. Others, Sultan, okay. Okay. So, second it is processing level. After processing, it should be transported from the nucleus to what it is cytoplasm. That is transportation level. So, gene expression exerted at transcription level, processing level. Third it is transport. So, what do you call this splicing? It is alternative splicing. What do you call it? Is alternative splicing. Okay. Third it is at transport level, that is mRNA from nucleus to cytoplasm. Transportation level. Its lifespan is going to be affected. It should be protected from the activity of some or it is other enzymes. So, in this lifespan of mRNA affected by many enzymes like exonucleases. So RNA has to be protected from the activity of exonucleases in order to express for the synthesis of what it is the protein. Here exonucleases, suppose this is mRNA. What it is? Cap. what it is, tail. Suppose it is exonuclease. It is 5 end, 3 end. Which are ready to clear mRNA, but it is prevented from this. Without capping, without tailing, RNA is going to made into what it is, the fragments. So, lifespan of mRNA affected by many enzymes like exonucleases. So, it should be protected from such activity of exonuclease by capping and tailing. Processing, that is transportation level. Transported mRNA protected from this, transported from the nucleus to cytoplasm. It is going to be translated. So, translational level. So, protein undergoes modification. Addition or deletion of the groups depends upon the requirement to become a suitable protein. So, protein undergo modification. Protein undergo what it is? Modification. That is addition or deletion of the group. This is level. So, transcriptional level, processing level, transportation level, and translation level. So, how many levels? 4 levels. Gene expression in eukaryote exerted at 4 levels. Transcription, then it is processing, then transportation, then it is translational level. So introduction okay. Got it? Priya, Himalakshmi, Uma Sultan, Kurshida, Lucy, Vamshidhar Reddy. Okay. Okay. Or writing the notes, very, very important, because certain sentences are given, we are elaborating those sentences. Okay. Okay. Then and gene expression regulated environment, metabolic, physiological. As earlier said, genes provide potentiality, environment provide varieties, opportunity. Okay. Suppose if you take E. coli. E. coli, for sake of energy source, it use varieties the glucose. Use glucose. Okay. Which catabolize energy it is available. E. coli, in absence of glucose and in presence of lactose, disaccharide, made available in medium, surrounding medium. Disaccharide made available in the medium. It is glucose is absent. In absence of the glucose, if lactose is available, genes switched on for lactose metabolism. Genes switched on for what it is? Lactose metabolism. So, lactose enter into cell. Now genes switched on. To hydrolyze lactose into glucose plus galactose. Again the galactose is converted into what it is, the glucose. So glucose utilized source of what it is, energy then. So the genes are switched on whenever there is a requirement. Physiologically it requires what it is, energy. Metabolical activities require, it is energy. And environment you are providing what it is, the lactose. So then genes switched on. So genes switched on and off depends upon the requirement. Okay. So in this, lactose is a disaccharide. It should hydrolyze into glucose and galactose. So E. coli as a source of, for sake of energy, it depends upon only glucose. In absence of the glucose, only it is lactose. So glucose catabolized energy available, it is a general thing. And if glucose not available, lactose made available, then genes switched on to catabolize what it is, lactose, to break down into glucose and galactose. Okay. So gene expression regulated by environment, metabolic, as well as what it is, physiological level. As earlier we said, if plant having TT, capital T, capital T, so certainly it has to exhibit what it is, tall character. But environment, due to some reasons, that is, environment is not favorable, temperature it is not favorable, and some other factors, nutritional factors. At that condition, even though it is having potentiality, the genes having potentiality, it is not supposed to express its character. As genes are providing potentiality, but environment is not providing that opportunity to express what it is, the genes. Here, environment is providing what it is, opportunity for expression of the gene we supplied. Anyway, in medium, lactose is available, the gene switched on. So, gene switched on and switched off. It's a great mechanism. Unnecessarily, genes are not going to be turned on, because whenever glucose is available, even though lactose is available, you do not, genes are not switched on, only for what it is, glucose catabolism only, but not for what it is, lactose. So even the microbe, it do not spend on energy unnecessarily. Whenever it is required, it makes. Do not required, it do not makes. Because everything is an energy expensive process. Transcription, it is energy expensive. Translation is also energy expensive. So whenever required only, it attempt to do what it is such metabolical activity. So microbes are so clever in conserving the energy. Are you getting it? Microbes are so clever in conserving energies because as it is energy expensive, not required, it is not opting for what it is genes to be switched on. Genes always to be switched off in such conditions. Okay. Okay, environ. Anush Koka, okay. Next, all. Okay. Vanshida Reddy, okay. Next, very, very important topic. So, are ahead of what it is the syllabus, and we can revise number of times as we required as far as the molecular biology concerned. Modi Lucy, Sheikh, okay. Vijay, okay. Arun Teja, okay. Others, rest of the members, okay, it is gene regulation, prokaryote, eukaryote, transcription, initiation, it's a predominant target site for control of the gene expression, okay. Okay. Okay, Pawan Sagar, okay. While at the time of login, initially, you message your name as well as variety section. Okay. Pawan Sagar, okay. Okay. So that is the gene regulation. So metabolic, environmental, metabolic as well as variety physiological. This is one point. And embryo develop into adult organism by undergoing so many number of the changes. Controlled and coordinated expression of various genes. If any sort of defect in expression of the gene, there is abnormality in case of what it is human being. If the gene is not properly expressed, no proper enzyme. So the hereditary diseases, because of what it is, the deficiency of what it is particular enzyme for particular metabolical activity. So genetic diseases, lack of particular metabolism, lack of particular metabolism, no enzyme to regulate. No enzyme to regulate means genes not properly expressed. Genes not expressed, like that you have to understand. Okay. So gene expression by synthesizing the protein. Okay. All you can leave the class. All you can leave the class.